This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 231. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today from Small Things Considered, Elio Schaechter.
Well, hello there. How are you doing?
How's it going? You still locked down, Elio?
Absolutely, you bet.
Good. Stay safe.
Did you manage to go to Los Angeles for Thanksgiving, or did you hunker down in San Diego?
No, we stayed home.
Smart.
Also joining us from Ann Arbor, Michigan, Michele Swanson.
Hello. What a treat to see you all.
Are you actually in Ann Arbor or out in the woods?
I am. I'm in Ann Arbor, college town. It's quiet, though.
You got some snow the other day, right?
We did, but it was a decorative snow. It doesn't stick to the roads or sidewalks, just nicely on the grass and bushes. So it's lovely.
And from Charleston, South Carolina, Michael Schmidt.
Hello, everyone.
Who we will forgive from being from a red state.
I am from a very red state in a city of blue.
Yeah, you're in a
city of blue.
Our guest today is, he's in a blue state for sure. He is from the University of Puget Sound in Tacoma, Washington. Mark Martin, welcome back.
Hi there. It's so nice to see the Twimmers again.
Yeah, it's great.
By the way, it is 45 degrees here in Tacoma, 7 degrees Celsius.
All right. You don't have any snow, do you?
Not yet.
You will.
We get like 3 days. We get 3 days.
So Mark is back for his 4th visit. How about that? That's pretty good.
I'm delighted to be here.
Tell us what you have on the wall behind you, Mark.
Oh, well, my wife, Dr. Jennifer Quinn, and I do a lot of Lux art. And so there you have a painting of the world done in luminous bacteria.
Nice. Very nice.
I always change these backgrounds back and forth. So I'm sorry if that's disturbing to people, but it's fun.
Not disturbing whatsoever. If you like what we do here on TWiM, consider supporting us. Show your love for microbiology, microbe.tv/contribute. Now, over on TWiV, we had a little space theme a few weeks ago. I don't know if any of you saw. We were able to interview Kate Rubins while she was on the International Space Station. We actually recorded live. It was awesome. Although,
wild.
2 to 3 second delay is a little weird, so you can't really have a spontaneous conversation. But I must say thank you to NASA. And today,
so keep those donations coming. It wasn't cheap to get Vincent into the space shuttle up there.
Well, he also had Condit with him, too. Condit was the expensive one.
Yeah, we had to limit it to 2. Anyway, that was a lot of fun. And, they're very open to doing those sorts of things, and that's great. So, they have a busy schedule, and it's not, you don't assume that you just should be able to do it. So thank you very much, NASA. Thank you, Kate Rubins. And, maybe one day we can do one here on TWiM. Anyway,
be lovely.
The reason I
guess
say this,
those of us who are feeling maybe a little bit claustrophobic with the shutdown, imagine being on the space shuttle. So I'll bet they appreciated some fresh. Space stations, fresh voices.
You have to be used to small spaces. I've been looking at old photos of Mercury and Gemini capsules. Man, you talk about small. Ooh. You got to sit there for a long time, right?
Yeah.
That requires a certain kind of person that can just sit there. You're told to sit there, just sit.
And now you know how John Glenn was able to withstand the United States Senate all those years.
You bet. You bet. All right. So in honor of space, Elio's going to give us a little snippet that's out of space. I already used that before.
It is completely out of space. In fact, the title, this is a paper in Nature Communications. And the title gives it away, Space Station Biomining Experiment Demonstrates Rare Earth Elements Extraction in Microgravity and Mars Gravity. Now, this characteristically is a paper which has 30 authors. I will not mention the names except the first one, who I think is very senior, Charles Cockle from Edinburgh, and the last one is René Demetz. And they are from all over the place. This is all over Europe. It includes Great Britain, Holland, Belgium, Germany, Italy, the United States. I won't read the rest of the names. So anyhow, here's the story. It turns out, as if you think about it, that if you want to know what happens in space, it ain't easy. So that what is a relatively trivial experiment on Earth requires a space station. But luckily, this is what the space station is good for. It's a platform out in space with no gravity to speak of, or very little gravity, and the ability to do experiments. And sure enough, they do experiments galore. This one is about whether you can duplicate in space what you can readily do on Earth, namely extract rare earth elements from minerals. Now, rare earth elements, as many of the listeners probably would know this, are terribly important in the electronic industry in making alloys. This is what you need them for all kinds of things. It's just life as we know it today would not be possible because they are involved in making microprocessors, making all kinds of things.
And just to drive that point home, cell phones, computer screens, and other high-tech applications are totally dependent on this class.
And one of the things we go back to our high school chemistry, most of the rare earths are in the lanthanide series of the periodic table. And if you think of the lanthanide series of the periodic table as Russian dolls, they are nested into one another. So you open the first and then but it's backward Russian dolls. You open the one that's of the lesser atomic weight, and as you build up, so you get the bigger Russian dolls. And what's remarkable about the lanthanide is they have an unfilled F sublevel of their electron shell. And they have a tendency to fill before the inner levels of the electrons fill. And this is what probably makes them very attractive to microbes. The one thing you need to appreciate is that these are metals, and microbes love metals because they exploit them to make energy and do all sorts of other things. But that's wandering off topic, and I'm going to get smacked.
You are going to get smacked because you said the things I was going to say. But that's okay.
I'm sorry.
Anyhow, sure enough, as Michael said, mining by microbes is common. In fact, if you're looking at your wedding ring, it's gold. That gold was probably extracted using microbes from minerals. And so they are important. In fact, microbes are essential in biomining, bioleaching, it's also known as. And so if you have a mineral and you want to extract the metal that you want from it, you use microbes. Using basalt as the source of the possible rare earth metals, as I say, rare earth metals are essential. In fact, we got a problem because one of them is we're almost done mining it. There's almost not going to be anything left. And then what are we going to do? I don't know, but it's going to be a problem. Anyhow, but the question that they ask is, you can extract these minerals using bacteria on Earth. Can you do it in space? And why would you want to know that? Well, you want to know it because you may want to build electronic equipment in space someday, or on another planet, or in somewhere else other than on Earth, right? So if you need to know under what gravity conditions do you do this. So instead of mimicking some lower gravity on Earth, they said, we have a space station, let's go to the space station and do it up there. This is what this little paper is about. And the conclusion, by the way, is that, yes, it works as long as you use one kind of bacterium, Sphingomonas dessicabilis. That's the name of the bug. If you use Bacillus subtilis or another mining favorite, Cupro avidus, they don't work very well. They don't work any. They do something on Earth, and they don't do any more than that on space. But the Sphingomonas, that's fine. And with using the Sphingomonas, sure enough, they were able to extract some of the lanthanides. Not all are the same, but it's good enough for this purpose. So anyhow, they find that using the Sphingomonas, they can extract a lot of the rare earth metals, some more than others, vanadium, lutetium, lanthanum. They have all very funny names, by the way. And so that's the way it works. So they even know a little bit about how it works. The bacteria raise the pH, and raising the pH is good for extracting the minerals. That's about it. So in a way, it's funny because you're asking not a trivial but a simple question, and to do it in outer space, you got a problem. By the way, they use 3 different kinds of gravities. They mimic the Earth gravity. Of course, they do the same experiment on land as well. They use a Mars-like gravity, and they use practically no gravity. So under these 3 conditions, they can determine that Sphingomonas is a useful organism. And someday, if you want to play this game and extract rare earth metal elements someplace else, you better use Sphingomonas dessicans. Give or take, that's about it.
I thought it was interesting that the bacteria appeared to just solubilize these rare metals from the basalt rather than take it up and use it in their own as a building block.
That's right. And it's not quite clear why they do it, but these Sphingomonas can do it. That's right.
And the other curious thing going along those lines that Michelle's bringing out is that Sphingomonas makes a large amount of this extracellular polysaccharide. That's and right. Any of you who have been looking at microbial mining or looking at novel microbes, you know that this EPS compounds that many microbes make is extremely important to the mining industry. And so if we begin to think about why a microbe is doing this sort of behavior for us, it's really to effectively do something for it. And the it, we just don't know. We can speculate, but we just don't know. And the authors offer us some of their speculation, talking about leaching and other things. But it's rather fascinating to begin to think why a microbe would begin to mineralize a rock like basalt, which has all sorts of things in it that then may be deleterious to the microbe, but the microbe has figured out a way to make its living, if you will, from this behavior.
One of the side products of this work is that they figure that this biomineralization by organisms like Sphingomonas may be a biomarker, in the sense that if you find this activity elsewhere, it would be an possible indication that there is life there, because this is a living thing, Sphingomonas. So it doesn't have to be Sphingomonas, it has to be something that does it the same way. Not a bad thought, but I think it's far-fetched a little bit.
And one of the things you may wish to know is the individual who was doing the experiments on the space station, Luca Paramitano, is actually one of the authors of the Nature paper. So he's sort of buried in the middle of the author.
Why is that surprising?
Yeah, he should be an author.
Yeah, he better be.
Why not?
Well, he meets all the criteria that nature sets up for being an author. He did the experiment. He was present when it was done because no one else was on that space station with him.
I like the name of this. They have a chamber basically where they put the rock and they put the bacteria in the chamber, right? And it's called a bio-rock experimental unit.
Yeah, it's quite complicated.
It sounds very NASA-like.
I didn't make a point of that, but it's really quite complicated. It's not easy to do these experiments and do them at different gravities.
Yeah,
and to get it controlled properly. I mean, that's really the remarkable bit. And that's one of the highlights of their paper. They talk about their non-biological control, and they share the data from the non-biological control versus the biological control. And you can see the differences of what you see is actually occurring in space, in the Mars gravity and the Earth gravity.
I speaking, of experiments in space, here's my question. Do they fail as often as they do on Earth?
Absolutely, I'm sure.
Or more. Who knows, right?
Elio, I wanted to tell you that a few years ago, I had one of my undergraduate students do some kind of mission in northern Mexico, and he found this dried up saline type of lake. And he brought me back, and I'll have to send you a picture of it. It's a shell covered by what I am quite sure is manganese.
Really?
Yeah, and it just deposited manganese all over the shell. And
I seem to remember that, yes.
Yeah. And so this idea about changing the redox state of a metal to change its solubility is something that really blows everyone away. And it's good to know that it happens on other planets too.
Well, it happens on the space station.
Right. Well, we'll get there.
We'll get there.
I won't, but someone will.
All right. Thank you, Elio. And speaking of
my pleasure,
blowing you away, now Mark Martin's going to do that, right?
Yeah.
Sure. I have a long-term interest in predatory microbes. I have my notes up here, and
there's nothing predatory. There's nothing predatory about yourself, is there, Mike?
Not for many years, Elio. We don't talk about that period of my life in my 20s. But what I was going to say is that one of the things that I tell students is that science progresses by a series of stages. Stage number 1 is that's kind of a dumb idea. Stage number 2 is, well, it might work a little bit in the lab, but it's not relevant to the natural world. And stage 3 is, I said it was a good idea all along. And a research student of mine who's now an OBGYN in Los Angeles, one of the best research students I ever had, even though she doesn't really want to continue in science, she's doing wonderful jobs as a physician. But she actually put it a different way. She added a 4th one to it and said, and the 4th is, and I came up with the idea first. And we've all seen this over and over again.
My idea.
And I remember when bacteriophages were presented to me as something that weren't terribly important to ecology. And then by the time I was in graduate school, maybe in a couple of areas it would be important, but not generally speaking. And now we know they're drivers of diversity, is certainly in the marine environment, and I'm sure everywhere else. And I am certain we're going to be there with predatory bacteria and predatory archaeans as well. And so what I like about this is that I first got interested in this whole topic because of a 1986 paper by Guerrera and Lynn Margulis in the Proceedings of the National Academy of Sciences. And it really influenced me a lot. And basically was the description of several different types of predatory microbes identified observationally. And there's the Delavibrio, my dear friend, which tends to invade between the inner and the outer membrane of gram-negatives, called the periplasm. By the way, Elio, you'll love the fact that I once said periplasmic space to Terry Beveridge. And he said, put out your hand, and he slapped my hand and said, it is an electron-dense gel. So the periplasm is not a space. And I don't slap hands, but I make that a big deal. So Delavibrios use that approach. Then we have something that called Daptobacter that I would love to see someone actually grow. but there may be a solution to that based on this paper. And then the third is the topic for today, the aptly named Vampirococcus. And I simply adore these things. And I have a Lynn Margulis story later if you're interested, because I only spoke with her on the phone once. I know she was a friend of yours, Elio.
She was.
But the title of this paper, and it's a preprint, is Reductive Evolution and Unique Infection in Feeding Mode of the CPR, and that always makes me laugh, the CPR predatory bacteria Vampirococcus lugosii.
And CPR, Mark, you need to probably explain to our generalist audience, it's not cardiopulmonary resuscitation. It's candidate phyla radiation.
That's right. I was going to get there.
Okay.
But in fact, they had me at lugosii because, I certainly love that kind of thing. So I was charmed instantly. Because I am not elegant and well-spoken, I'm going to attempt the last names of the authors because I'm so happy to see the way in which they wrote and what they found out. Morera, Zibanagic, Lopez Arquia, Inesto, and Lopez Garcia. And they're in France and in Spain. And Spain, as anyone who looks into this knows, has a long history of looking at predatory microbes. And one of the things that I've long been interested in is the diversity in predatory style. I mean, going back to the idea of Martin Dworkin with Myxobacters showing this wolf pack idea where they're secreting hydrolytic enzymes in front of the advancing rank of the group bacteria. And there even was one, and I know Elio's written about it for Small Things Considered, Lewin writing about ixotrophy among the Saprospira, which is where you entrap things in flagella. Fact is, it's not simply about the idea of Della-Vibrio ramming into a cell and invading the periplasm. Furthermore, there's lots and lots of examples of bacteria riding on other bacteria. And then the question becomes whether they're hitchhikers or freeloaders. And that's one of the things that I started thinking about. There is, for example, a type of a relative of Della-Vibrio that attaches epibiotically. But the big mystery was, what about these uncultivatable predators? Because I believe predators are very, very common in the microbial world. And that's what I like about what they did here. I need to tell you that I have an expression I use with my students, and it's #OMG for overwhelming microbial greatness. And this is a good example of it right here. As Michael told you, CPR stands for candidate phyla radiation. And I used to think of that as the way that they used to describe the pseudomonads, as this big catch-all group. But the fact is, candidate phyla radiation includes a lot of different things. And one of the members is this fascinating organism called Saccharobacteria. And it's within the phylum. And I've got to tell you, they also charm me with this, and I hope I pronounce it properly because I love it when I see it. Phylum Abscondidibacteria. So absconding with things. I just love that idea. And the Saccharobacteria we know are predators, and there's some interesting work done about that in Boston. But what this paper did is they took advantage of what they see observationally, that they were able to identify some type of predator-like organism attaching to and appearing to feed upon an anoxygenic gamma proteobacterium, probably Halochromatium. And these hitchhikers sit on it. And then what I love about this is they used micromanipulators to get some of those cells. And then they were able to do single-cell genomics. And that way they were able to figure out what was there with only two possible sources, the prey, for lack of a better word, and the predator. And this reminds me so much of the work that Carl Stetter's group did with Ignococcus and Nanoarchaeum.
Oh, yes.
But now things have improved so much more. So this mixture of optical and electron microscopy was used to identify what appeared to be predatory events taking place. And then the single cell genomics took them there. Now, I have to tell you, I have a little bit of a prejudice, and I want to be open with everybody about it. because part of it is my encroaching age. I have difficulty with structural genomics. And the reason I do is there aren't really experiments there. They're just descriptive. And you will only learn about things that are related to other things that you know. And if I may be allowed this digression, when many years ago I got a chance to review a manuscript about the genome of Vibrio, there was a portion of it that was very interesting to me. The authors said that many people had hypothesized that Vibrio was stealing ATP from prey cells. But because the genome sequence didn't show any relatives of ATP translocases from chlamydia or rickettsia, this couldn't be true. And I looked at that and I said, I don't know. I can think of 3 papers by Ned Ruby showing more than one ATP translocase biochemically in Vibrio. And that really makes me very cautious about structural genomics because we can't really do the experiments. So in any event, they were able to kind of identify what they believe is the predatory organism that they've given this charming name to. These are epibionts that attach to the surface of this halochromatum like a prey, and they form stacks. And the photographs are so fascinating in this paper to show these stacks. Now, these are tiny. These predators are 500 nanometers basically in diameter and 250 nanometers tall, and they can pass through 0.2 micron filters. They appear to be forming stalks, although the photographs that I saw, I'm not really sure that I'm seeing them as clearly as they are. So I'll take their word for it on that one. But this business, are you not believing? Are you laughing at me there, Vincent?
No, I agree with you. Yeah.
Yeah, I used to get into this a little bit with Pili. But in any event, certainly there's something attaching to them, forming these stacks. I agree with that. And there's some description that they make about forming stalks. And all of that, I thought, okay, maybe how much of that is artifactual? I can't answer that question. But what is absolutely true is there appears to be damage taking place to the cytoplasm of the prey organism, and the predator is actually gaining its living from that. And what my notes tell me, that is to say, tell me, is that this is a really interesting predator. Again, very similar to the Saccharobacter that I mentioned before. A reduced genome, just over 1 million bases, missing so many biosynthetic genes. What I found interesting is that apparently they had trouble recovering the, I gotta get the name right, the halochromation. They had trouble getting the halochromation genome out of it. And they believe that's because nucleases were kind of chewing it down during the process. Personally, I would have looked for other members of the halochromation and got its genome separately, but I wasn't there.
But you grow these organisms in vitro so that you could add radionuclide precursors and then measure the halochromatia radioactive proteins going into the predator species.
This would be wonderful, but you can't grow them. You can observe them.
So therein goes to, we have to figure out how to grow the halochromatium.
And I looked at this and I thought to myself, if we could use nanosims, we could even kind of watch certain nutrients being transferred. Although they say from TEM, they're not seeing attachment structures. But they see this, they call it kind of an ornate rugose. I love their language in this whole paper. Outer surface of it, which is, I thought, very interesting. Michele had a question, though.
Yeah, you emphasize that there just aren't that many genes. And it's not just that they don't have many genes. They're missing entire standard pathways, like making a protein, making lipids, like membrane and protein.
I need to back down. I'm getting too excited because
and yet they're alive.
I really like this.
Tell us, how does this work?
Because I remember when Schaechter first reported nanoarchaea, a lot of people thought these were just little blebs that contained some material. They had to actually show there was a genome there, and that took some time to do. So on the path that takes you from something that is a commensal or a parasite all the way to something that becomes an organelle, you're going to be losing things as time goes on. One of the big mysteries to me with Delovibrio is how large the genome was. I would have expected this Darwinowing to take place over time, but I'm not seeing it here. But you're absolutely right, Michele, missing all kinds of things that you wouldn't expect. I love the description of ATPases. It does have an ATPase, thank goodness, but it doesn't have an electron transport system. Now, you and I can look at this and you can say, can you really be sure of that just based on the genome stuff? This is, again, my prejudice. And there might be ways to test that. There are some very clever people with certain, I want to say biochemical, and I mean more redox dye techniques to actually look. For example, is there a proton motive force there? Because absolutely anaerobes can run ATPase backwards to generate proton motive force to do all kinds of things. So I think this is just a fabulous thing that they've identified here. I thought it was interesting that we don't see any kind of attachment structure, though, Michele. And I had
maybe
right. Thank you, Elio.
Their membrane is certainly decorated with a lot of large proteins, but their purpose here was not to study the function of those yet.
And I don't know how easy it would be to do that with this system is what I'm getting at. It may be, I can't guarantee this, it's a relative of Saccharobacter, and you might be able to use that as a chassis to kind of see if you can get some of these Vampirococcus genes expressed in there, maybe. But I agree with you 100%. These giant proteins in particular, which they believe seem to be part of this very thick layer, which didn't look so thick to me, but I don't do a lot of electron microscopy, was fascinating to me. And it made me think a little bit about myxobacters. So this idea with the myxobacters is they're making all these hydrolytic enzymes in front of them, yet they're still getting benefits, even though there should be diffusion in all directions, not just the wolf pack approaching. So I'm wondering whether or not all of that excess exterior to the cell, the outer wall elaborations, actually act as a way of just keeping the stuff in a relatively confined area so that they can just be essentially sucked up because there isn't an actual touching business taking place between predator and prey.
Mm-hmm. Yeah, and I really appreciated the schematic that they included in their preprint that kind of gave us a conceptual framework to think about what they've discovered in the genome and then in the microscope. Because now you can do lots of hypothesis-driven research if that's the avenue they want to pursue.
Oh, yeah. It's really interesting to me. They say there's some evidence of horizontal gene transfer. And this is something that triggered me for 25 years ago, because I was certain we would see all kinds of horizontal gene transfer in Delovibrio. There isn't a lot of evidence for that. I thought sure it would have picked up, certainly the ones that I was familiar with that Sid Rittenberg had been growing on E. coli for 25 years before they got deposited at ATCC. I thought sure we would find some IS elements common to E. coli in there, never been seen in Delovibrio. But there is some evidence that things have been transferred in there because of GC differences. And so we're getting a lot of good hints to things from this. The other thing that they went crazy about here is apparently a lot of the people in this group work on CRISPR systems. And so to a carpenter, every problem is solved with a hammer and nails. And to people who like CRISPR, they want to look for CRISPR, which is fine. And they found CRISPR and they found restriction modification systems and all kinds of things that blew my mind, because I'm thinking you don't have an electron transport chain, but you do have restriction mod systems. And that just seemed very strange to me.
That suggests that there is nucleic acid exchange because they don't want to, tinker with the secret sauce recipe.
I think that's exactly right. I mean, how you maintain your genomic integrity is a big issue that you would think predators would need to worry about if they had motivations.
But even a first step before the integrity of your genome, you've got to be able to make your genome. And they found none of the standard enzymes that make nucleotides, the building blocks. And so the idea is they're scavenging those too. They're using these CRISPR enzymes and such and restriction modification enzymes to chew up the DNA they're sucking from the host cell that they're attached to.
We better have pentose phosphate pathway is all I can say.
So let's ask a Vincent question. Is this thing a virus, since it is unable to replicate its genome because it has none of the building blocks? It's unable to make protein. Is this thing a virus, Mark?
And it's filterable.
It's filterable.
Yeah.
Which is the original definition.
It's the original operational definition. Vincent has been sort of sphinx-like through this discussion.
How does it divide? That's the question. Does it divide by binary fission or by assembly of components? And I'm willing to bet it's by binary fission, right?
And it probably has FTSZ.
It also has peptidoglycan.
Yes.
So it's not a virus. But
it could be a sophisticated virus.
Not a virus. No, it's got that binary bit, which no virus does. Even the biggest, giantest amoebal viruses, they don't do binary fission. But I wanted to ask Mark, is this a parasite? You call them predatory, which means they eat things, right? But are they parasites? It seems to me that they are, right?
That's such an interesting question. And Elio will remember Mortimer Starr. And
am I the repository of all memory here?
You are. You are.
Mortimer Starr, who I never actually met at UC Davis, wrote a 25-page review trying to define if Delavibrio was a parasite, a parasitoid, predator. And it was all very interesting to read, but I could never get those 3 hours back. So to answer your question, Vincent, what I would say to you is I think this is on the path to being a parasite, because I don't really see this as being quite as modal and capable of independence. Let me explain what I mean. You can find naturally occurring mutants of Delavibrio that can grow on nutrient media. So that you can do. Mm-hmm. So they can grow independently. True, they're doing that on rich media, but they are able to grow. We can have a discussion about what changes have taken place another time. But with this, I think just like nanoarchaeum, I cannot imagine this living on its own. You could bring up the Mycoplasma genitalium issue. Is that an independent organism because it needs so much? At the same time, it is able to reproduce in a fashion, seems to have more of the essential genes. But this is almost like an organelle that you would see in a eukaryotic cell, is what I was thinking.
So my other question is, are there viruses that infect it?
Now, I'm almost certain there are. And I hope they can, and I don't know how you would find out with this, but maybe looking in the CRISPR system to see in the repeats if you have the fingerprints of what was left behind. Yep. I do know that there are some Holin genes in there. Are there not, Michele? You were deep-oriented. And that makes me wonder whether or not they were from prior phage attack over evolutionary time after Darwin filed off the serial numbers. Right? That's what
Darwin files off the serial numbers, show title.
That's great.
That's what I tell people about genomic islands, is that they've just, been eroding, right? So we don't really see where they're from. So I think that's the case. I know, because Henry Williams published on this, that there's a virus that goes after a relative of Delavibrio. So you have a predator, and then you have a parasite or a pathogen on that predator. I would not be at all surprised. I do think that the people doing work with Saccharobacter, which again is part of the oral microbiota, would be the best place to look for that. But honestly, I'm almost to the point of saying, if you look in the repeat sequences of the CRISPR systems, can you find evidence of other types of phage-like proteins there?
Yeah, I mean, that's, of course, limited by our database of phage sequences. We don't have everything for sure. And so we have to wait to get that. But yeah, that's the good way to do it.
So you said you had a good story about Lynn Margulis.
Oh, absolutely.
What was that about?
So when I first returned to academia from working in biotech, and I was recently engaged to the woman who's now been my wife for over 20 years. Her father was a physicist, a theoretical physicist, and her mother told my wife, never marry an experimentalist because you'll never see them. And so it was, I had written to Lynn Margulis because she didn't have email. This was in the middle 1990s. And so I sent her a letter. I talked about the PNAS paper that I sent you, and I wanted to talk a little bit about modes of predation by bacteria, stealing energy, things of that nature. And it was about 7:00 on a Sunday night, and I was late for dinner. I only had a few more experiments to do. And the phone rang in my office, which at that time was attached to the lab. And I go, and I just know it's Jenny, and I know she's mad at me, you know. So I pick up the phone. Hello? And this voice I don't recognize says, but you're working late. And I look at the phone and I say, and you're calling late. I'm Mark Martin. Who are you? And she goes, I'm Lynn Margulis. And she started talking to me. And I shouldn't say to me as much as at me, because I'm not being critical. I was taking notes furiously. And at one point, I just threw my pen over my shoulder because I wasn't going to be able to keep up. It was like from the fire hose. Do you know what I mean, Elio? She was going so fast and there was so much there.
Do I know what you mean?
Yeah.
Yes.
And she went from topic to topic to topic to topic. She was very insistent that we just needed to know the right way to grow Daptobacter and that it was everywhere. Ditto with Vampirococcus. She thought Delavibrio got too much play, all that kind of stuff. I listened as much as I could, but she was just so authoritative but charming.
Mm-hmm. And if it was 7:00 at night on the West Coast, it was very late on the East Coast on a Sunday night.
Yeah. So she had so many interesting ideas, and I miss the rabble rousing that she did. Yeah,
it's a good story. Very, very descriptive.
So Lynn Margulis's work was known to this team that I was able to correspond with the lead, David Morara. So he is the co-director of the DEAM team. So DEAM, their Diversity, Ecology, and Evolution of Microbes group that he co-directs at University in Orsay, which is about southwest of Paris. So their goal there is to study different aspects of diversity and evolution in both prokaryote and eukaryotic microbes. So this has this interest in these not blood-sucking, but cell-sucking microbes.
Cytoplasm.
Was actually a side project, but they happened to be doing some sampling in this lake in Spain of the microbial mats. And it was at that time that they had just purchased a really great inverted microscope that had the micromanipulator attached. So it was the first time, this project was the first time they were able to take these little parasitic cells and suck them up into the glass capillary. And as he sat there doing this painstaking work, he knew, like, this is it. This was the technical hurdle. Now we can apply our really sophisticated genome and phylogenetic analysis to start to get at some of the secrets. And it was during one of those late night sessions that he realized that they had to name this species the lugosii, because he too was a fan of the Dracula 1931 film, a classic. So they are interested, again, this was a serendipitous discovery. They weren't looking for it, but because they were familiar with Lynn's work, they were able to put two and two together. And by the end of this project, they realized what an interesting group of microorganisms these are. And so they've decided to put more of their effort to understanding them as a representative of these sea PR bacteria. So they're going to start looking for them in some of the freshwater ecosystems that they've been studying for many years. So David did his PhD work in phylogeny and genetics, studying acidophils in the Autonomous University of Madrid. And then he moved to Paris and did a postdoc studying molecular phylogeny of protists, and then earned a permanent position with the French Research Council, known as CNRS, and was able to co-found and co-direct this DEAM team group where he now works. So he does have some advice for junior colleagues. He says, try to work on questions that really grab you, because it's the best and maybe the only way to keep the motivation and enthusiasm alive during the really painstaking hours that we all have to put in to crack the code of our problems. He also says that most of the work that their team does is really sequence analysis, bioinformatics, phylogeny, but they try to combine it with field work, because he's convinced that any good bioinformatician is going to do better work if they actually know where the organism came from, and then they can think more creatively as they're doing their really sophisticated DNA sequence analysis. So when David is not doing this amazing work, he also enjoys reading, gardening, and classical music.
Thank you, Michele.
Yeah.
Michele, can I say that I can tell that I would like this person just the style in which he wrote?
Yep. It was hard for me not to interrupt at the very beginning when you were talking about the micromanipulator. I imagine the two of you would have great conversations.
No, I mean, his you can tell he loves it and he's enthusiastic, and he uses language that is so far removed from speaking in a sterile fashion. I understand it's not for everybody, but I applaud it.
Yeah. They do have a website, the deamteam.fr.en, and I see that they have open positions. So if you're interested, if you're a motivated graduate student or a postdoc interested in this type of work, give them contact them.
There you go, Elio.
And
Absolutely.
And I see that they recently did another field trip, December of last year, to a lake in Chile, high altitudes of Chile. So it's a really interesting combination of field work in exotic places and the really sophisticated genomic analysis.
All right. I have to say that when Mark sent this paper to me a few weeks ago, and he said, if you ever want to do this paper, I'm on it. And I said, okay. You're on it. Thank you, Mark. That was great.
Very welcome.
I have a couple of emails I would like to read. One from Heather, who writes, Dear TWiMians, a great episode as always. Regarding the letter discussed at the end on poisonous mushrooms, or rather regarding the following talk, I can maybe add a very little. So I think the question was, what was the question?
Why are mushrooms toxic?
Why they make poison?
I think it was something more than that. But anyway, you get
who does it benefit?
It was almost a Talmudic question, right?
What's in it? What's in it for the mushroom?
Yeah.
What's in it?
Because there's such a delay before you get sick, so it doesn't seem like it's well designed to shoo away predators.
Here it is. Is it possible that mammals became poisonous to mushrooms, not mushrooms became poisonous to us? Okay. All right. Elio is quite right that mycologists have been discussing the why of mushroom poisons forever. And this was the subsequent discussion. You're absolutely right. Why do we have toxins? And with very little in the way of a solid answer regarding the deadliest known mushroom poisons, amatoxins, these are produced by mushrooms in 4 unrelated genera and act by binding tightly, though not quite irreversibly, to eukaryotic RNA polymerase II, halting transcription and ultimately protein synthesis. They are actually not effective against mammals, they are not effective against bacterial RNA polymerases, and in eukaryotes, more effective against mammals than insects, against insects than plants, against plants than some single-celled organisms like trypanosomes, which are unaffected by alpha-amanitin and other amatoxins. As discussed, this is not likely to have been selected as a defense compound against humans or mammals due to the delay in symptom development. 2 possibilities are that amanitin is protecting against insect larvae, Drosophila is susceptible to amatoxin poisoning, but resistant Drosophila with mutations in the amatoxin-binding residues of RNA polymerase II have been selected in culture, or against mycoparasites. Hypomyces hyalinus parasitizes edible Amanita mushrooms in the section Validae, but has not been found parasitizing mushrooms in the amatoxin-producing sister section Phalloideae. So it might be effective against low-motility organisms living in close contact with the mushrooms over prolonged time.
I find that very pleasing, a pleasing explanation.
Not a bad idea.
So
yeah.
I like that.
Heather is an assistant professor of practice in food science and technology at the University of Nebraska, Lincoln.
And she knows a lot about mushrooms, which makes it dear to my heart.
And one more from Hibiscus. Hello, Vincent and company. I'm a microbiology student in Southern California. The weather is starting to give us need for electric blankets and heaters at night, being between 70 and 49 Fahrenheit, 22 to 9 C. Extremely cold for natives of sunny California who think of snow as a legend.
Toughen up. That's great weather.
I've been listening. You leave all the windows open at that weather.
I've been listening to both TWiM and TWiV on and off for a year or so. I appreciate all what I have learned about viruses and microbes. Your discussions are very entertaining and insightful, and my study sessions are that much more enjoyable. My first introduction to microbiology was through a manga or Japanese comic called Moyashimon. This manga is about the shenanigans people at Agricultural University get up to, but the interesting part is the main character who can see microbes without a microscope. While reading, I found out that one of my favorite authors was a microbiologist, Beatrix Potter, the creator of Peter Rabbit and Jemima Puddle Duck. I was wondering how often her paper was discussed in microbiology classes or whether she has been lost to obscurity. Thank you for reading.
That true?
Hibiscus. I don't know. That's what Hibiscus says. Do you know, Mark?
Do you discuss Beatrice Potter, Mark?
I do not, but I did want to say that the manga series, which is called Tales of Agriculture, not only can this person see microbes, but he can talk to them.
Ooh.
And let me tell you, I want that job.
You'd like to talk with him, too. I think you'd get along.
Mark, if you could talk to a microbe, what would you say?
Why?
Yeah. The universal question.
So many questions.
Oh, no.
How?
My students tease me so often because I'll talk about the motivations of a microbe. And of course, they have none other than the fact that they're older. I mean, I'm wearing...
No, they want to make two microbes. That's their motivation.
Just first evolved, last extinct, right? What more do we need to know?
Exactly. Okay. That is TWiM 231. You can find it at microbe.tv/TWiM if you want to send us your story or a question. TWiM, TWIM at microbe.tv. If you would like to support us financially, you would send it to microbe.tv/contribute. Let me say that again. If you'd like to support us financially, please go to microbe.tv/contribute. Our guest today, Mark Martin. He's at the University of Puget Sound. Thank you, Mark. Great to see you.
Always a pleasure.
Who's the statue on the shelf to your right there? The tall one. Who is that?
Oh, well, that's Aldous Huxley.
I can't quite see. Of course.
Yeah. I have Aldous Huxley and I have George Orwell here. Come on.
Right. I should have known. I'm sorry. My apologies. Michele Swanson's at the University of Michigan. Thanks, Michele.
Thank you. And everybody, buckle down. We've got a couple rough months ahead, but we'll get through it together.
Elio Schaechter's at Small Things Considered. Thank you, Elio.
And thank you so much, Mark, for joining us. It was a real pleasure.
Thank you, Elio.
Michael Schmidt's at the Medical University of South Carolina. Thank you, Michael.
Thanks, everyone. Enjoyed it.
I'm Vincent Racaniello. You can find me at virology.blog. I'd like to thank the American Society for Microbiology for their support of TWiM, and Ronald Jenkins for the music. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.